Hello everyone. In this video we're going to talk about a few of the food tests that you do at key stage 3 and 4. So we're going to talk about the one for starch, the one for fat, for glucose and for protein. So we've got this nice plate of food in the middle. You can see it's full of pastas, breads. What else do we have there? Looks like we've got some wheat, we've got some potatoes. But let's think about whether any of these particular products contain the food groups that we're looking at today. Now, starch is a large carbohydrate that can be broken down into sugars like glucose, which you use for energy. You use fats for energy, for protection, for warmth and insulation. So we've said already that glucose is used for energy and protein is used for growth and repair of cells and body tissues. So that's why we need these particular food groups. Let's talk about testing samples for them. So let's start with starch first of all. So we're going to talk about the test for starch. Now, the first thing to do would be to take a small piece of crushed up food that you want to test and put it into the well of what's called the dimple tile. So you might have seen these little tiles. If I just draw them from a bird's eye view, here's a little tile with little circles on that look like this, empty wells. And that is called the dimple tile. So the first thing you want to do is add food sample to essentially a dimple tile. So you place it there. Then we want to use a chemical called iodine. Now iodine will turn bluey black if starch is present. So the test for starch is to use something called iodine. Now iodine goes black with starch. We'll write in here, use iodine, it goes a black colour if present. So if starch is present, the iodine will turn bluey black. So that's the test for starch, quite simply. Let's look at another. Let's look at fat. So now we're going to take one of our food samples and we're going to see if there is any fat present. Now this one is perhaps even simpler. At A level, when you test for fats, you do something called the emulsion test. Now, I've spoke about the emulsion test in my video entitled The Role of Lipids or Fats and the Emulsion Test. So it's called the emulsion test. Now, that's a more specific food test, but at key stage 3, this is what you would do. So this video is aimed at key stage 3 and I'm talking about what you do at that level. So at key stage 3, you would simply just rub a small piece of the food, so rub a small piece of your food sample that you're looking at over paper. That's all you do, just rub it over some paper. Now, if the page goes translucent, so that's almost sort of see-through if you like, it almost goes greasy looking, it's called translucent. So if it goes translucent, lets a little bit of light through, then we say that fat is present. Now at key stage 3, that is the test for fat. Just for completeness sake, I will just mention briefly about the emulsion test. In the emulsion test, you actually take the sample and dissolve it almost in ethanol. So you place it in ethanol for a few minutes, then in water, and you get this sort of milky white precipitate forming. And that would show the presence of lipid or fat. But that you'd only really talk about at key stage 4 actually and A level. This is a key stage 3 particular video. So we've done starch, we've done fat. Let's look at the two big ones, glucose and protein. So for this we're going to shrink the screen just a little bit. So now we're testing for glucose. And protein. And I've put them from the same sort of red line because they start the same. They both have the same method to begin with. So you want to mash up just a small amount of food and an equal volume of water using a pestle and mortar. I'll just mash up, I'll put here, mash up food and water, but I'll draw so it looks like a bowl. So this is our pestle, and then you've got something that looks a bit like this, the mortar. So we mash up with equal volume of both, the equivalent amount of water for the food that we have. Then we'd want to take that and transfer some of that food-water mix into a tube. So we transfer some food and the water mix to, and we can just use a simple test tube. So we'll put it to a test tube. That's the second thing you do when testing for glucose and protein. Now this is where it becomes a bit different. For glucose, so we shrink this down just ever so slightly more. For the glucose, what we then do is add 10 drops of something called Benedict's solution. So we need to add 10 drops, roughly 10 drops, of what's called Benedict's. Once we've added 10 drops of Benedict's, we then want to place that tube in a beaker of water and heat it over a flame. So we add Benedict's and then place the tube in beaker of water and heat. And if glucose is present, then we could get an array of colours, but usually it's greeny yellow, or even red colours can indicate that glucose is present. Benedict's is a sort of bluey solution, so if that turns greeny yellow or red, then glucose is present. So I'll put here, in brackets next to Benedict, turns, I'll put green/red. So that's key. When we test for glucose, we're using Benedict's reagent. Now, sometimes that gets confused with protein, because protein uses something also beginning with B, but not Benedict's. This time it uses something called B-array. So when we're testing for protein, we actually add to that test tube of food and water something called, I'll write it in red as I did Benedict's blue. We add what's called B-array solution. Now B-array, or the B-array solution, is actually made up of sodium hydroxide solution and copper sulfate solution. So you can even make this up yourself in a lab. 10 drops of sodium hydroxide solution with 10 drops of copper sulfate solution will make what we call a B-array solution. So if we add 10 drops of the B-array solution to our water food mix in the test tube and wait a couple of minutes, then it should turn purple, and that's indicative of protein in the food. So I'll even write this in purple. So we add the B-array solution and it turns purple if protein is present, and that's a test for protein. So there we have it, our 4 key stage 3 tests. For starch, we're using iodine that goes black. For fat, we're going to rub it on paper and see if it goes translucent. For glucose, we're going to mash up the food with some water, add 10 drops of Benedict's reagent, place the tube in a beaker of water and heat. And if the Benedict's turns green or red, then we know that we have glucose present. And for protein, we're going to mix that food and water again, but this time I had a BRX solution made up of sodium hydroxide and copper sulfate solutions. So we can do that ourselves. Wait a couple of minutes, and if it turns purple, then there's protein in the food. So there are 4 basic food tests. Okay, I hope that helps.